So welcome. I'm Arturo Dominguez. I'm the head of science education. Um, I know several of you were expecting Andrew, and so I am here to fill his shoes, hopefully. Let me see. I'm going to start here. Welcome back. Yes, for everybody that's here, we know this is just our second talk here in main campus. I know most of you are used to going to PBPL, but except for the coffee, we brought the donuts and bagels and juices. So, they're yes, the winner, the champions, yes. And I saw more mugs today, so I think people learned their lesson from last week and brought their coffee. So we adapt. We adapt. Um, so just going a little bit through the logistics. For in-person participants, since the pandemic, masks are optional and welcome. Um, there is, I believe, a room for overflow. I think today we are doing okay. It's not the cafeteria. It's, I believe, upstairs in the mezzanine. We will have our speaker give the presentation, and then we'll hold the questions until the end. I'll moderate the questions. But if what we've done in the past, if there's urgent questions, if you're
That's fine with me.
okay, then you can address them. But just keep in mind that because of Zoom, all questions have to be given microphone, so there will be some lag time there. So if you can hold your questions until the end, that would be best. For Zoom participants, the Microsoft should be set to mute. Please make sure that is done. And also, we will take your questions at the end. We'll try to keep track of your questions as they come in. So a little bit. So you're here, so I guess I don't have to direct you to Mater Hall. Just a reminder: no food or drink in this auditorium, including coffee. So please, if you have coffee or drinks or food, please keep them outside. And today's speaker is Professor Bonnie Firestein. She's a professor of cell biology and neuroscience at Rutgers University. She's an associate editor for Science Advances and for Molecular and Cellular Neuroscience. Sorry. Professor Firestein is a fellow of the American Institute of Medical and Biological Engineering and the American Association for the Advancement of Science. She started her lab in 2000 after earning her doctoral degree from the University of California, San Diego in neuroscience and performing her postdoc work at UC San Francisco. In addition to her research in schizophrenia, Professor Firestein has identified drug targets for traumatic brain injury and spinal cord injury-induced pain. She was president of the New Jersey chapter of the Society of Neuroscience from 2009 to 2020 and is active in outreach to the community. We love to hear that. So Professor Firestein knows about the three questions, and so we will begin. So what got you excited to get into science, to get into the work that you do?
So I've always been interested in science, and to my parents' dismay, I never wanted to be a medical doctor. I run the opposite way. And so I was really torn between whether I wanted to be a mathematician or whether I wanted to be a biologist. And when I was an undergraduate at the University of Michigan, this is when neuroscience was a new field, and it was called biological psychiatry. I took a course in biological psychiatry, and I decided neuroscience is the way I want to go. But I've always been interested in science, and my parents actually sold our house in Huntington, New York, to a pretty famous scientist. So when I came home from junior high school, my dad's like, "You're going to be so excited. He's a scientist." So apparently, I always wanted to be a scientist.
That's awesome. Yes. And so while you were developing your interest in the field, was there a professor or a teacher that really, when you think back, were essential in this process?
So when I was in 10th grade, my parents moved me to Florida. And since we're in New Jersey, I grew up in New York and was really unhappy going to Florida. But it was a brand new high school, and I had this amazing math teacher who was teaching us also how to code. And at that time, I'm pretty old, so at that time we were doing things like BASIC and Apple Machine Language. So they were Apples, they weren't even Macs. And he was brilliant. And he actually went to Michigan. That's part of the reason I got inspired to go to Michigan. But he really inspired me. In fact, I sent him an email a few years ago and said, "I just want to let you know, so many years ago, you were an amazing teacher." And I was very lucky, very fortunate.
That's awesome. Okay. So I just read through your really impressive bio. When you're not doing all of this really impressive work, what do you do in your free time?
So I do CrossFit. I used to go to a CrossFit box, and now my husband and I do it at home. So I love to work out, and I love to spend time with my husband and son. And we have 3 dogs. We have a miniature dachshund and 2 beagles. So we watch a lot of old movies, and
Oh, that sounds
we all sit
amazing.
on the couch.
That sounds amazing. Before I ask y'all to welcome Professor Firestein, since I have these slides open, I want to just take a quick second and advertise next week's talk, the final talk of the series, The Science and Art of Polling by Patrick Murray. So with that, why don't we switch over to you and let's welcome Professor Bonnie Firestein for today's talk. Thank you for being here.
I think so, yes. So thank you so much for inviting me to come. Thank you for coming to my talk. I think it's very, very important as scientists that we show you where your tax money is going. So I've always wanted to be a scientist, but my main goal is to really help people. And I really want to help people with neuropsychiatric disorders, as well as people who've suffered injuries. So today I'm going to tell you a little bit about the work that's going on in my lab about schizophrenia and give you a broad idea of the types of work that people do to try to help these patients. And I call my talk Schizophrenia, a journey beyond the brain, because towards the end, I'm going to tell you a little bit about our work identifying biomarkers. And you can't take pieces of brain from people who are living. We need to find other tissues in the body to try to figure out how to diagnose these people and how to treat these people. Okay, why is it not going forward? We'll try this then. It is not going forward. There we go. Okay, just needed to see. Yep, so we're good to go. Thank you. So dendrites are important for receiving information. When you look at nerve cells or you look at the brain, there are multiple cell types. There are nerve cells, which we call neurons. There are astrocytes, which are supporting cells. There are oligodendrocytes, which wrap our neurons full of myelin, which is this fatty substance, so that they can perpetuate signals. But we really focus a lot on the neurons. So shown here is a schematic of 2 neurons that are communicating. The first neuron is called the presynaptic neuron, and I'll explain to you in a minute why. We see a cell body, and then out of this cell body grows these branches or these tree-like projections. These tree-like projections are called dendrites, and they receive information, which then is integrated in the cell body. There's also an output center called the axon. This generally looks like a piece of spaghetti. Some of them are pretty smooth, but some have these little arbors at the end. And this axon is the output center. So there's a second neuron here, which is shown. It's called the postsynaptic neuron. And when this neuron wants to communicate with this neuron, it releases chemicals called neurotransmitters at this region called the synapse. So this neuron says, hey, I want to signal this next neuron to learn or to remember something. It will release neurotransmitters, and maybe some of you are familiar with glutamate or serotonin or dopamine. It will release some of those chemicals. And then on the dendrite side of this postsynaptic neuron, there are receptors or proteins that bind these chemicals, and then it tells this neuron, hey, propagate a signal. So this neuron can tell this neuron now to be active. And so what's important about this is the shape of these dendrites tells the neurons how to receive information. So you can imagine some of these dendrites look like large trees, large arbors, and some look short and stubby like bushes. And if you think about the cell body as kind of our core, right, depending on if your arms are long or your arms are short, I always tell my students, if you pinch my arm, if it's further away from my body, it's going to take a while to get to my body, where if it's short and stubby, you're going to feel it much more quickly. So it's the same thing with dendrites. These dendrites, if they're long versus if they're stubby, they're going to process information differently. And that's why different neurons have different functions. So what is some of the problems with neurodegenerative diseases or neurocognitive diseases? What happens is a lot of these dendrites are pruned off or they're lost. So this is a schematic drawn by a former postdoc of mine of a normal neuron. You see lots and lots of dendrite branches. And I'm not going to talk about this today, but these branches sometimes are not smooth. they have little stubby projections that stick out. These are called dendritic spines. They look like little mushrooms. And this is actually where information goes in. But not all neurons have these spines, so I don't want to talk too much about them today. But what I want to show you is in all different types of disorders, so autism spectrum disorder, Rett syndrome, and schizophrenia, you can see that there are a lot less dendrites. Okay. And we're going to focus on schizophrenia today. Alzheimer's disease, although the neurons start out normal, they end up losing their dendrites as people age, as well as Fragile X syndrome, Down syndrome. And this is another key, stress and anxiety, which I always worry about because we're all stressed all the time, so our dendrites are going to look pretty bad. So today I want to focus on schizophrenia. The difference between schizophrenia and Alzheimer's disease is schizophrenia actually is set up in the womb. So it's not that you're going to get schizophrenia, but you have a risk. And one of the risks is having less dendrites. So today I'm going to talk a little bit about what might make a decrease in these dendrites and what some of the outcomes might be. Why schizophrenia? It's a serious psychiatric disorder that affects about 1% of the population. New estimates, when corrected, come down a little bit less than 1%, but it's still about 1%. Genetic basis has been established through studying family members, which we'll talk about in a minute. So schizophrenia, it's not like you have a mutation or you have a problem with one gene that encodes one protein. So I could have schizophrenia, one of you could have schizophrenia, and the genetic basis could be different, even though we have similar symptoms. And that's because it's affecting pathways of proteins and the way neurons talk to each other. So it's a very, very difficult disease to treat because no two people are exactly alike unless they're identical twins. But even with that, environment plays a factor. So what do I mean by that? Let's start at the bottom here. In the red bar, this is identical twins. So identical twins were studied, and they found that if one of the twins had schizophrenia, there's a 50% chance of the other twin having schizophrenia. So what does this tell us? There is a genetic link, but there's also an environmental link, because if it was only genetics, this bar would be at 100%. If you start moving up, where you're looking at first-degree relatives, this decreases to about half. So if you look at fraternal twins, which share about 50% of their genes or DNA, it goes down to about 20%. Children of someone with schizophrenia goes down to 13%. Parents and siblings are below 10%. And as you move up to second degree, you see a decrease. And then the first degree cousins and general population decreases. So this tells us there's a genetic component, but again, that's not the full story. And I show this picture not to make you dizzy, but just to give you an idea of all of the genes and all of the proteins that have been linked to schizophrenia. So if you look here, there are genes and proteins that are actually even in peripheral nerves, not even in the brain, outside of the brain. There are proteins and genes in microvessels, so the blood vessels in the brain. There are genes at the synapse, both in the cells that I told you wrap the axons of neurons. There are proteins in neurons. There are proteins in these support cells called astrocytes. And here's the neuron itself. And so what I wanted to show you is we focus on one of these proteins. You can see the one arrow here. And we really study it in detail. And I'll tell you in a minute why. But this is just to show you that even though we're focused on this protein with the red arrow, there are so many other proteins that need to be studied. And there are other labs working on them as well. So what are some of the symptoms of patients with schizophrenia? To be diagnosed with schizophrenia, you need to have at least one symptom in each of these domains: positive domain, negative domain, and cognitive domain. So when we think positive, I think excitement, happy. But actually, for a patient with schizophrenia, this is a negative thing. So we call them positive symptoms because it's something that they've acquired that control or normal people do not have. So for example, hearing voices, seeing things, having disorganized speech, that's not something that a normal subject has, right? So they have something added to their normal functioning. Negative is something that's missing. So flat affect. So what is flat affect? So if I tell you a joke, hopefully you'll laugh. It depends how funny I am, but in general, you'll laugh. A person with schizophrenia is not going to laugh. If you tell them something sad, they're not going to cry. They're going to be very flat. And that is one of the negative symptoms. They have decreased speech, so lack of proper speech. They get social withdrawal. So even introverts, like my husband, will go out and interact with people. But patients with schizophrenia do not want to have any contact really with the outside world. Avolition, so they lose their motivation. They don't want to do anything. And loss of enjoyment. So I would say, I couldn't imagine life without chocolate, and I can't imagine losing my joy for chocolate. These people lose their joy for anything from chocolate to basic activities. So those are the negative. And then cognitive is what you think of as learning and memory. So executive functions, higher order function like reasoning. Working memory, they have problems remembering things. Attention, they can't pay attention. And then lastly, decision making, they have a horrible time making decisions. So again, to be diagnosed, you have to have at least one symptom from the 3 domains. So we're going to focus on that one little protein that I showed you. Again, we study other proteins as well, but today, for the sake of time, we're going to focus on this one protein. And so what we find are differences in the DNA of patients with schizophrenia versus neurotypical individuals from multiple populations, such as Canadian, Han Chinese, Colombian. There is also a Swedish cohort from Minnesota. They have changes in their DNA in a gene called NOS1AP. And these differences have been linked to schizophrenia. So a change in this DNA doesn't cause you to have schizophrenia. It gives you a higher risk. So for example, if you have a traumatic event, if you have changes in your DNA, you're more likely to develop schizophrenia. Doesn't guarantee that you get it. So I'll tell you today a little bit about the study of schizophrenia. We're going to go from cells all the way up to humans. And I think that each step of the way, we learn a little bit more about how this protein and other proteins as well are involved in schizophrenia. So my laboratory's research is on a protein, which is NOS1AP, that's expressed at higher levels in patients with schizophrenia than without. And I'm going to tell you a little bit about what the consequences of this are. So the first thing I want to tell you before I show you some data is that there are 3 different sizes of this protein, NOS1AP, that I keep harping on. It's not important, but just know when I show you data in a minute that because there are 3, the question is, are all 3 of them changed in the brains of patients with schizophrenia? And this protein is related to learning and memory. So we and others have studied it, and it's very important. So it gives us an idea of how changes to this protein might disrupt learning and memory. What we did is we took postmortem brain tissue from patients with schizophrenia and control patients. We didn't actually take it. We got it from a biobank. And we looked at the protein levels of the 3 isoforms or 3 sizes of this protein NOS1AP. So what's shown here along the bottom is the longest isoform, the short isoform, and the even shortest isoform, so the different sizes. What's shown on the left here is the amount of this protein, NOS1AP. It's normalized to a protein that tells us how much we've loaded. That's what GAPDH is. And I want you to notice that each ticker is 10-fold higher. So normally when you see axes, they count from 1 to 10. Here we're on a log scale. So if you move up 1 notch, you've actually gone up 10-fold in the amount of protein. Let's look here. So the gray bars are patients with schizophrenia. The white bars are control patients. And you can see in these postmortem brain samples, they're in the cortex, which is important for learning and memory and is involved in schizophrenia. It's on the outside or top of your brain. You can see that there's at least a 10-fold increase in the amount of this protein. For short and the very short isoform, it's even more dramatic. So the question is, okay, what are the consequences of overexpressing? Because we know that if we increase the levels of NOS1AP, potentially we could increase these symptoms of schizophrenia. So how did we study what the consequences are? These are pictures of mice that either have too much NOS1AP or more NOS1AP in their cortices, just like these postmortem brain samples, or have control levels. Tag RFP is just to control. These are normal animals. NOS1AP means that they have too much. What the postdoc did is you can look at sections of brains and stain them and fill the neurons, and then you can draw the neurons by hand. This is called neurolucida drawing. What you can see on the top here are the drawings of the neurons that he observed under the microscope, and on the bottom are the neurons that he observed in mice that have too much NOS1AP in their brain. And from these pictures, you can see there are a lot less dendrites, right? These guys have maybe one or two, maybe some of them are lucky enough to have a few more, but they definitely look completely different. And as scientists, we always love to quantitate. So we counted either adding up all of the dendrite lengths together. So if we add up every single dendrite length, we see that there's a significant decrease in the dendrite length when there's too much NOS1AP. And if we count how many branches, we see that there's a significant decrease. So you can imagine if there are patients who are in the womb, they might develop schizophrenia later on in life, and they're more at risk for schizophrenia. Maybe they have less dendrites. And we'll talk about that in a minute. The other problem in these mice is that their neurons are in the wrong place. And this has also been seen in patients with schizophrenia. So while these mice are developing, what's shown on the left here is just a control. So there's a normal mouse. Some of the neurons are highlighted in red. On the right is a mouse that has too much of this protein, again, in its cortex. And as a human or a mouse or a rat develops, when they form the cortex, neurons will migrate out and start forming the cortex. So they start kind of in the center of the brain and migrate out. They stop migrating, they grow dendrites, they grow an axon, and they start connecting together. What happens when you have too much NOS1AP? So here's your control mouse. It's kind of hard to see, but you can see that there are 3 neurons that are in the middle of the brain, but most of the neurons have migrated out to the top, right, top of the cortex. But when we overexpress NOS1AP, you can see there are a lot more neurons that are stuck in the middle of the brain. And so again, we'd like to quantitate. This is the percentage of neurons that did not migrate, that are stuck in the middle. And there's a significant increase when NOS1AP is overexpressed. So you would say, okay, that's great. You just showed me data. You're a scientist. What does that mean? And why is that relevant to patients with schizophrenia? And the reason is when patients have gone in for MRIs and scientists have studied what the connections between the regions of their brains are, they've seen these differences. So for example, let's go to the top panel here. The red lines are these connections between brain regions that are stronger or more, there are more of them in patients that are controls versus schizophrenia. If you go down to the second panel, it's the same thing. These red lines connect different regions that are stronger or there are more of them in patients who are control, or I should say subjects who are normal versus patients with schizophrenia. And then lastly, this is the reverse. So in green are those connections that are stronger or different in those with schizophrenia than control. So you can see that you're now disrupting the entire brain. So what are the data that I just showed you? How are they relevant to this? Well, if you have less dendrites, you have less communication between neurons. If the neurons are put in the wrong regions in the brain, you're going to have misconnections just like we see with patients with schizophrenia. And again, I just want to remind you, it's not cut and dry because a lot of these changes, these huge, massive changes, are not only due to the one protein I'm telling you about, they're due to multiple proteins, they're due to different experiences that cause psychoses or cause negative and positive and cognitive problems. The other important thing about schizophrenia is that there are gender effects. So the incidence of schizophrenia from men to women, it's 40% higher in males, and we still don't know why. So often when we recruit patients, there are more men than women. Additionally, men, the onset of their symptoms is about 4 years earlier than in women, and they're usually diagnosed in late adolescence to early 20s. The thing about women is that there are factors, psychosocial factors, that are often found in menopausal and postmenopausal women that make any symptoms of schizophrenia worse. So women might go through depression, they have hormonal changes, and so there are some gender effects later on in life. And then lastly, the biggest problem is that men and women have different responses to treatment. And actually, it's an even bigger problem than that because individual people have different responses to treatment because the genetic basis and the environmental basis for their schizophrenia is not the same as someone else's. So this is a really big problem. So we asked a very simple question. We said, what if we just take some normal animals and we inject them with 2 different antipsychotic medications that are on the market right now, and we look at the differences between males and females? Are they affected differently? We looked at 3 different proteins, including NOS1AP, to see if they change differently in males and females. So I will focus on NOS1AP, but I just wanted to show you. Here's NOS1AP. DISC1 is disrupted in schizophrenia. It's another gene that's involved. And then the dopamine 2 receptor. We took male and female rats, and we injected them with D-serine, which is one of the drugs on the market. We did it once a day for 12 days, and then we analyzed their proteins. And what's interesting is in the males, NOS1AP went down. And this isn't in mice with schizophrenia. This is just regular old, rats, control rats. DISC1 went down, and the receptor was untouched. If you look at the females, what I really want to highlight, besides the fact that you get this opposite for DISC1, is that NOS1AP doesn't change at all. So you can imagine if NOS1AP is increased in someone's brain, maybe if they're male, the best course of action is D-serine, but not for females. Because maybe the D-serine will reduce the NOS1AP in the males, but not the females. So then we ask the question, okay, maybe let's try another antipsychotic medication that's been around longer, haloperidol. We did the same experiment for 12 days. We injected haloperidol, and we looked at these proteins. And unfortunately, haloperidol had no effect in males or females on NOS1AP. In fact, it had a very small effect overall. So this would tell us that if the basis for someone's schizophrenia is too much NOS1AP, haloperidol is probably not the right treatment, whether you're male or female. So in summary, there's overexpression of NOS1AP during neuronal development, decreases the number of dendrites. Overexpression also results in improper positioning of neurons in the cortex. And those two together can explain why during development, why the brain is set up with improper connectivity. Why in brains with patients with schizophrenia, eventually, their connectivity is different. And the other issue is that environment also influences how your brain is connected. So when you learn something, it might change the connectivity if you have a traumatic event. So if you already have neurons in the wrong place and you're at risk for schizophrenia, now you might even have a bigger problem because now you kind of have started, instead of at point 0, you've now maybe started at point 10, right? Where you're already have this disorganization in connectivity. And furthermore, there's a sex-dependent effect of antipsychotics on proteins, including NOS1AP, that are linked to schizophrenia. So one of the problems is trying to figure out what are the best strategies to treat patients with schizophrenia, not only based on their genetic basis, but also on whether they're male or female, and if for females, if they're postmenopausal or premenopausal. So the next part I want to tell you about is how can we help diagnose or give a prognosis to patients with schizophrenia? This is kind of the end of the basic science portion. Does anybody have a burning question before I move to humans? So the question is, we saw a sex-dependent, make sure I repeat the question properly, a sex-dependent change in these proteins when we treated with antipsychotics. And the question is, with the female rats or humans, do we see? Yeah, so they were grouped here, but we did not see sex-dependent differences. So, and again, this is a general population, so there is a wide variation because they were just from a brain bank, but we did not see any sex-dependent differences at that point. Anybody else have any questions? Impact of brain injury. So that's considered one of the traumatic events. If you have risk for schizophrenia, it can make it worse. There are other problems, and we think that's environmental, not just the injury itself. It also depends on where the injury is, but often people with traumatic brain injuries have post-traumatic stress disorder, and that can trigger psychosis. Oh, and I'm sorry, the question was, how about people who have had an injury? Does that, let's say, increase the risk of schizophrenia? The answer is yes.
I have a quick question, actually. At the very beginning, when you showed the rates of schizophrenia for fraternal twins and siblings were different.
Yes, they were,
And
which is odd, right? So,
right? But like,
yes.
okay.
So, right. So the question is, why are fraternal twins at 17% and, for example, siblings at 9%? Nobody really knows why. You could argue that fraternal twins have similar experiences where, right, because they often go to school together and they're often hanging out together. And so possibly it could be the environment, because this would tell you it really actually has to be the environment. It can't be genetics. I think somebody asked something on Zoom. Let me see. I'm terrible with the chat on Zoom.
I'll try to open it and keep track from now on.
I can never find where the chat is, and I don't want to mess this up. Maybe we can answer that question in a bit.
Yeah, let's hold it till the end.
Okay. Oh. So if Halidol has been given to a female, I can't see what the rest of it says. Sorry. Okay, let's go to. So we then decided we were going to work with patients with schizophrenia. And this was a big move in my lab. What we did is we recruited patients, and I'll tell you a little bit about them, and we tried to look for biomarkers. We want to figure out a way that we can not have to go into someone's brain, right? That's kind of ridiculous, where we could either diagnose them or prognose them, give them prognosis. Now think about when you have the flu. So this is the best example. My husband actually gave me this example. You have the flu, you go in and you say to your doctor, I think I have the flu. And your doctor says, well, great, we're going to give you this test and we're going to look to see if you really do have the flu. It's objective, the test says yes or no, and you're good to go. You can be treated, right? They can give you Tamiflu, or I think that's what they give. Well, with schizophrenia, the problem is you can't do that. Right now, there's no test. And the only ways patients with schizophrenia are diagnosed generally is that they go into a psychiatrist's office, they go through extensive questions and extensive testing, and then the psychiatrists diagnose them. And even though psychiatrists have seen many, many, many of these cases, each case is different. It's a subjective diagnosis. And also, schizophrenia is a spectrum. It's very similar to autism, where there's schizophrenia, there's schizoaffective disorder. So we would really like a way to be able to say, you walk into the doctor's office, they have a test, or they send it out to LabCorp or Quest or wherever, and they say, yes, you have schizophrenia, or no, you don't. And these are the best medications for you to take. So we can tailor your medication. So this is, of course, not completely finished, but it's in process. What are some of the biomarkers that we can look for? So a biomarker comes in different categories. The ones in red are the ones I'm kind of addressing. So one type of biomarker is to look at susceptibility or risk. And this, for example, if we wanted to do biomarkers, kind of cumbersome, but you could sequence the DNA and see if there are specific changes to the DNA. It's not a super quick test. What would be nice is to have one where you could diagnose. So looking at a specific protein, looking at a specific mRNA, which is a precursor to a protein, that would be great. We want to be able to monitor, so we want a biomarker that changes as somebody is taking medication, that you can kind of keep track to see if it's getting better. We want something that'll give a prognosis. So patients with schizophrenia will have times where they're kind of in a refractory period where they might not have as many symptoms. So we call them, they have different episodes. So in between the episodes, they seem to be relatively normal, and then they have another episode. It would be nice to be able to figure out if they're going to have another episode, which is prognostic and predictive. And then we want to see if they have a response to medication. And then lastly, most drug companies will look for biomarkers for safety. So what would be very nice is if we could find a biomarker or a set of biomarkers that can go from diagnosing all the way to seeing if there's a drug response or medication response. So how do we do this? So I really hate needles, and I was talking to my student, and I said, I know people often do pinpricks in the finger to collect blood and look for biomarkers. I don't like that. You can do that for the first set of experiments, but I think you should probably take cheek cells. So it sounds like a very odd idea, but think about it. Would you rather have your cheek swabbed, or would you rather have, maybe some of you like needles? I don't, so I'd rather have my cheek swabbed. And it turns out that cheek cells come from the same we call it precursor cell, or the first cell that then becomes other cells. They share that similarity with neurons in the brain, which was great. I didn't realize it at the time, but it made it a really good set of cells to get. So what we would do is we would take the patients, and then we would age, gender, and race match controls, and we would take what we call a buccal, or some people call it a buccal swab. Those are just your cheek cells. It's different than saliva. It's the actual cells on the inside of the cheek. We then would isolate both proteins and RNA. So messenger RNA codes for proteins. So you can look one step first and say, hey, is there RNA in the cell? And eventually we'll make protein, or are the proteins there? And they will give you different answers. And then if we're looking at our messenger mRNA or our messenger RNA, we can do RT-qPCR. So most people know PCR, but it just amplifies. This is just a method to look to see how much of your mRNA is there for a specific, encoding a specific protein. And then lastly, we take these results and we compare them between control patients or control subjects and patients. So is there something different or between patients and subjects, control subjects? So we looked at NOS1AP again, because I can't give a 20-hour talk, but we said, okay, let's look at NOS1AP. This is that protein that I showed you, I've been talking about all along, that stops dendrites from growing. We asked, is there a change in the buccal cells? So if you take all 27 patients we collected and the control patients, and you put them together, or you analyze them together, it turns out that NOS1AP is not a very good biomarker right now for everybody. But what stood out is we have a very large South Asian population in our group. I shouldn't say very large, there were six people, and we were able to observe that NOS1AP mRNA levels are higher in cheek cells in patients with schizophrenia than control patients. And we were actually shocked that it worked so well. So this might be a biomarker for South Asian people who might have a risk of schizophrenia. It also might be a biomarker if they're given a medication. Does NOS1AP return to control levels? And we're working on we found some other mRNA levels and other proteins that actually affect, or I would say, represent the entire population. So we have a lot of different subpopulations, and then we're trying to split it so that we could have specific subpopulation biomarkers. And what's really important is, okay, we found that there's more NOS1AP in those cheek cells, but what does that mean? What's shown here is just one of many graphs. So this axis, the higher you go, the worse the symptoms are for patients with schizophrenia. So it could be positive symptoms. We see the same graph for negative symptoms and cognitive symptoms. And on the x-axis, we see how much NOS1AP mRNA there is, and you see this straight line. The more NOS1AP these patients have, the more positive symptoms they have, the more negative symptoms they have, and the worse they are at cognition. So this is a really great biomarker. We've even included the control patients, which are these black dots here. These are the patients with schizophrenia in red. So it's a really nice biomarker. We can predict if someone has more NOS1AP in their cheek cells, they would have a higher risk for schizophrenia, and they might even be experiencing some of these symptoms, all three of the symptoms. And then we can decide, okay, well, this person seems to have schizophrenia that's related to NOS1AP, maybe, and they're male, maybe we should treat them with D-serine. Oh, they're female, well, maybe we shouldn't use D-serine or haloperidol because it might not help. So in summary for this part, I have 5 more minutes, so I'll show you one other set. Patients with schizophrenia who are South Asian had significantly higher buccal cell NOS1AP mRNA expression than control subjects. And in our cohort, this expression was correlated with the presence and the severity, how bad it was, of positive symptoms, excitement, depression, disorganization, thought disturbance, and paranoia and belligerence, as well as flat affect and other working memory issues. So now NOS1AP mRNA from cheek cells can be a candidate for schizophrenia in at least Asian populations, maybe other populations as we get more patients, right? 27, it took a lot of work to get 27 patients, especially through COVID, but it's still a small cohort of people, right? It's still not as great a cohort. We need to represent more people. So for this part, we think that we've found a biomarker along with some other proteins that I don't have time to talk about, that now hopefully we can come up with either a test for the doctor's office where they can look at mRNA levels, or that they can send to LabCorp requests so you can have an answer within a few days. And now they can diagnose schizophrenia the way we do for the flu. So it's not based on what the psychiatrist thinks, it's a test that is objective. And again, the ultimate goal is to get this test to work. This is just in the beginning stages. I think I have about 5 minutes. So I'm just going to tell you a little bit about our cohort of patients and how we characterize them as being ill or severely ill. So patients come into the clinic and they've already been diagnosed with schizophrenia. And if they have not, let's say they're going through their first episode, there are psychiatrists on hand who then do the diagnosis. Again, we don't have biomarkers, so we have to rely on the psychiatrist giving a diagnosis. We then first recruited patients with schizophrenia or schizoaffective disorder, whoever came into the clinic. And after we tested them, we then recruited age, race, and gender match controls. Now, the reason this is important is that changes in the DNA that give you a risk for schizophrenia actually are different between different ethnic groups. So even when we talk about Caucasian groups, if you look in the database, most of the Caucasians who have data on them are really from England and Germany and Western Europe. They're not from Eastern Europe, and there seem to be differences. And you could extend that to other ethnicities, other races, right? So it's very, very important to make sure when you study schizophrenia that you have matched people. We then gave them 2 sessions. The first session, we're going to talk about in a minute, is called the SCID, and this is going to identify any major diagnoses. So the DSM-5 is a handbook that psychiatrists use to diagnose mental disorders or cognitive disorders. There are a bunch of symptoms, all of the doctors agree on it, and they publish this manual. The second test is called the SCI-PANS, and this is where in the past 2 weeks, it's self-reported, what are your clinical symptoms? So we gave them a structured interview for the DSM-5. Again, that's the manual that tells you what to look for schizophrenia. Again, we didn't always do this. We did this with the help of a psychiatrist. They had a semi-structured interview to determine any major diagnoses, so positive, negative, cognitive symptoms. It takes about 1 to 2 hours. So you can imagine we recruit a patient, they have to sit there 1 to 2 hours, and then we even have to bring them back for a second one. So we had a lot of people who left the study. Because remember, again, they have disorganized thought, they have attention disorders. We then gave them the SCI-PANS, which is the structured clinical interview for the positive and negative syndrome scale. They were given 30 itemed interviews to rate the extent of psychotic deficit and other symptoms. Again, they have to sit there for 1 to 2 hours. That's a lot. I don't even know if I would want to sit there for that long. And it takes 1 to 2 hours to administer. And this is just to show you don't have to really look at the numbers, but the point of this slide is that our patients range from moderately to markedly ill. So how did we detect that? These are just different types of parameters that we looked at: positive, negative cognitive symptoms, excitement, depression, disorganization, lack of motivation, thought disturbance, how long it takes them to actually activate, paranoia, and depression. And the only thing I want you to really take home from this slide is, if you look at patients with schizophrenia versus control patients, in every single one of these, the numbers are higher. And the higher the number, the worse you score on that scale. So our patients had always had worse scores than the control patients, and it's significant. So our cohort, unfortunately, was moderately to markedly ill. And so with that, I think that was 45 minutes. I just wanted to tell you a little bit about my lab. So I've trained graduate students, postdocs, undergraduate students. I've had high school students in my lab. I think it's really important to realize that I don't want to say give them the orders. They're a wonderful group of people, and they do all of the work. We discuss a lot of the work with them. We've collaborated with Stephen Silverstein and Linda Brustowitz, as well as Irving Vega and Jared Lamp. The past members, Kristen Crosta, did much of this work for her thesis. Kirsten Spain also did much of the work for her master's thesis. Again, I cannot stress enough that research is done as part of a group, not as a single person. And all of these ideas are important. So again, our work is federally funded, it's state funded. This work is not state funded, but other work is state funded. So your tax dollars and your speeding tickets, which go to brain injury and spinal cord injury research. So I would say keep on speeding, but I really don't want you to. They go towards training people because I'm not going to be around forever. I don't plan on retiring soon, but who knows? So we need people to carry on the work. And this is a picture of my lab from about 6 months ago. I want to thank the Rutgers Princeton Center for Computational Cognitive Neuropsychiatry. They funded this work. They actually gave me a small grant to recruit the patients. NIH, NSF, Brain Injury Commission, Spinal Cord Injury Commission, and this is the CCMP. So with that, I'm really happy to take questions, and thank you for listening. Please go ahead. You bring the microphone.
Yeah, what I'll do actually is I found the questions that were asked online Zoom, so
Okay.
we'll start with those, and then we'll go on to the audience questions. So online from a DT, how do you find if NOS1AP has decreased in people with residual schizophrenia?
So our thought is that you would take cheek cells, right? We can't go into the brain. So we would take cheek cells from these patients. And the thought is that if they're not experiencing these horrific symptoms, let's say they're in between episodes, the protein levels would go down. So then a doctor can ask the question, well, what medications are they on? What's helping? So the idea is that we would use, right now, nobody can do that, right? There's no assay, there's no test to see what the NOS1AP levels are. So that's what we're trying to develop.
Excellent. Yes. And I'll ask the second question. There's a question up front. Why don't we start with them? If Haldol, this was from Wendy, if Haldol has been given to a female for over 20 years of her life and ineffective according to NOS1AP, should treatment be switched to Deserine?
So that would be in consultation with your psychiatrist, right? So part of the problem is that, so if it's working, then we don't know what other proteins are at work. So maybe haloperidol, this person has problems with other proteins, so the haloperidol is helping them. But if the haloperidol is not helping them, then this might help to figure out what drugs to use.
It sounded like a very specific. So yeah, there was a question, you pointed to somebody here, and then we'll go. I know, yes, but the speaker has.
Oh, sorry.
We are surrounded by big pharma in New Jersey. How much of your work is a duplicate of what is being done in one of these commercial labs?
So we don't think that anybody's been looking at buccal cells. We don't know if anyone's developing biomarkers, like if I scour. So the way I look and see what people are doing is I look at PubMed, and it shows all of the locations. Like we also just, Rutgers submitted a patent application for this work, and they have to do a prior art search, meaning they have to see what other people have patented, and there was nothing like this. So, you know, I don't want to toot my own horn and say, oh, nobody in the world's doing it, but if they're doing it, I don't know about it because it's not out there in the public domain.
Excellent. The next question, yes? No, right behind you.
Yes. So it's interesting that I'm a proteomics guy. So for the NOS 1AP, are there sequence changes in the protein? And obviously there's a precursor protein that's a synthase for that protein. Is that changed? And have you seen epigenetic?
Yes, so we haven't looked at epigenetics yet, but when we did proteomics on these samples, and I didn't have time to talk about it, but NOS1AP did not come up. The change in the DNA for NOS1AP is in the promoter region. So for people who don't know, there's a region, so you have exons in your DNA, and that becomes your mRNA, and then it codes protein. But there are regions in between, and then there's a region kind of at the start of the gene that other proteins bind to, and it tells the gene to come on or come off. So all of they're called associated alleles, they're not really mutations. All of the associated alleles actually change the binding of transcription factors, so proteins, to the gene, and that's the way we think NOS1AP is increased. Now, it's possible there are epigenetic changes at that promoter, but we haven't looked at that yet.
Excellent. And yes? Yes.
Working? Yeah. Could you go back to your symptom slide?
Sure.
It was a wonderful talk, by the way.
Thank you.
Love hearing how the brain works.
There we go.
Okay. Those are not just the symptoms of schizophrenia. I could say the same thing for some age-related dementia. What's the relationship there? What do we need to know? What are we looking at?
So this is a problem because, for example, NOS1AP is also implicated in bipolar disorder. So I would argue that even worse than age-related dementia, there's bipolar disorder, which is kind of a sister, it's on that spectrum. So the issue is bipolar disorder is treated very differently. The medication you need is very different. And the hope is that, I mean, if in an ideal world I had tons of money to do this study, I would have recruited patients with dementia, patients with bipolar disorder. I mean, that will have to be done. I don't know if that's going to be done by my lab or if we finally get this technology going, pharma would do it. But that's a huge study. And you do have a really good point. So our goal, so this is not the only biomarker. So again, remember NOS1 for the Asian population. The general population, we saw another protein called SP4, which is a major hit for schizophrenia. So this protein SP4 binds to DNA for other genes, and then we find proteins that it regulates also are upregulated. So the hope is that you would see a set that's in common, and then sets of proteins or mRNAs that are different between the two. So that when we set up this, again, it's at the symbol stages, but if we set up this assay or this test for doctors or LabCorp requests, that they'll say, okay, there's overlap, but here are some unique proteins or mRNAs for schizophrenia. Here are some unique ones for dementia. Here are some unique ones for bipolar disorder, and be able to diagnose that way. And that's part of the problem, right? The psychiatrist says you have schizophrenia, but maybe you don't.
All right. Ivan, go ahead.
Are there any archaeological studies that can give us a timeline as to when the overexpression of NOS1AP or any other biomarkers began?
So nobody has really done that. I don't know if they've looked at when schizophrenia started developing. Part of the problem is diagnosing it. So it's things like you think about back, I don't know, even during like Kennedy time, Ethel Kennedy, right? They would lobotomize women who were hysterical. And so I think part of the issue is diagnosing what is schizophrenia and when was it first recorded. It's like Alzheimer's disease. Alzheimer's disease, I think, was reported in the early 1900s, if I'm not mistaken, by Dr. Alzheimer. And he had one patient who showed these symptoms, and he named it Alzheimer's disease. But this probably isn't the first person who ever had Alzheimer's disease. So I think that's part of the issue where we don't really know how to look, and there was no diagnosis until recently.
So I have a question about like the more subjective interviews done to diagnose patients like with schizophrenia. So since like a really prominent symptom of schizophrenia is like disorganized thought and like speech, how can we be sure how reliable their answers are for these interviews?
That's true. That's a great question, right? It's a problem because that's why we have a trained psychiatrist who makes the diagnoses. We never do. I mean, I'm not a psychologist or a psychiatrist, and my students certainly can't do it. It's not, so it is a problem, but I think the psychiatrists are trained to know there's disorganized thought, right? And sometimes the patients with schizophrenia have tics or they move differently. And so it's very, very subjective. I think that is part of the problem. You're getting really at the heart of the problem. It's a really good question. We have to rely on them, right? That's the best we can do right now. What there's some people are doing is these MRI studies to look at connectivity in the brain. But again, that's a biomarker, but it's expensive. It takes too much time. If someone's having a psychotic episode, you can't really wait. You can't even put them in an MRI machine, right? They're not going to want to sit still. So it is I don't want to call it a flawed system, but it's a very subjective system.
All right. Yes.
Interested. It's a really interesting talk. Thank you very much. Do you see opportunities to connect with smaller, more precise studies, larger public studies like UK Biobank? They're starting an initiative to do proteomics on the entire population. You've got that transdiagnosis. You don't have the precise psychiatric characterization, but just curious how you think these can interplay and learn from one another?
So I think for us, we had a very specific population, and we really wanted to be able to take a lot of their clinical data and correlate it with the molecular data. As biobank gets better, I don't know if they have buccal cells or cheek cells, but I could probably put in a request. And we're looking at buccal cells, but we don't know what would happen with skin cells or nasal epithelium or any of that. The only reason we chose buccal cells is A, because I just thought, oh, that'll be easy. Sometimes science is serendipity, right? And B, they really do have a common precursor, but maybe some of their data will be useful.
There's a question online from Prasad. Will the solution from Neuralink certain brain circuits help schizophrenia patients?
It's a good question. Based on some of the MRI data that I showed, you would assume yes. Part of the issue is knowing you'd have to activate and then inhibit different regions. Even though I said these are more active, that doesn't mean that the active pathway is not an inhibitory pathway, right? It does. You can have an activated pathway that then inhibits downstream. So I don't know. You would assume yes, but I don't really know. I think it's much more complicated, right? It has to be tested. I mean, the only thing I've seen is deep brain stimulation for Parkinson's. That works really well, but it's one region of the brain, and they know exactly what the heart of Parkinson's is, right? It's a loss of these specific neurons. So,
the next question is over here.
Yeah. Hi. I had a question. My question was, why is something which is happening in the brain observable in the cheek? Why would the cells which are being affected in the brain, the same thing be visible in the cheek? It's probably more a question of how the body works.
yeah, so it's a really good question. And why we think it is because the cheek cells came from the same precursor cells that the brain cells did, that the neurons came from. So maybe even though NOS1AP is going to function differently in cheek cells, right? Cheek cells don't have dendrites. There are other things NOS1AP does. So there's probably some regulation of the protein in cheek cells that's similar to the regulation in neurons. So remember, if your DNA has a change, it's going to be changed in your cheek cells and it's going to be changed in your brain. So if you now change the DNA so that the gene isn't transcribed as it's transcribed more highly, and then you have more protein, it might also happen in the cheek cells. I mean, it's a little more complicated than that, but because they're from the same precursor cell, we think that the proteins could be reflected. Not 100%, but maybe a subset of the proteins.
Has schizophrenia been seen in any animals, like non-human primates?
Has it been seen? It has not really been reported. People usually give drugs to induce psychoses. I haven't really seen anything about, oh, we have this subset of a population that we studied that has schizophrenia, but it could be because, first of all, primates are really, really expensive. So they probably have, usually if you look at a primate study, it's like 1 or 2 animals, and then they induce psychosis. But we try to do it in mice and mostly mice, but the problem is, how do you know if a mouse has schizophrenia? You can't ask it, do you hear extra squeaking? Right? Are your thoughts disorganized? I mean, we could do behavioral tests, but it's not really the same.
Where's the next question? Over there. I think we'll take this and 3 more questions. So next one is?
Hi. You mentioned that symptoms will ebb and flow over time. There could be episodes. Do you think that the dendrites are getting healthier and less healthier over time in patients? Are they active in that way, or is it just set and then that's it for your whole life?
So that's also a great question. So we don't know. Oh, the question is, you have episodes, and in between the episodes, people seem to get better. So do the dendrites go back to normal? And I think that was there more to the question? Yeah, do the dendrites go back to normal? And do they ebb and flow? So dendrites can grow, and they can grow when you have what we call an enriched environment, or if you exercise. So there's a lot about dendrites, making more dendrites, and that's a good thing. For patients with schizophrenia, I don't think that during that time they grow back all of their dendrites. I think it's not really possible when you're missing so many dendrites. It's a little more complicated problem than when you're developing. And besides the dendrites, if we go back super quickly, what could be happening, possibly, is these little spines that I didn't have time to talk about, they're very flexible, they're very plastic, and they move, they kind of come out, they come back into the dendrite. So it's possible that during those times, maybe these little dendritic spines change, and maybe they help. But nobody really knows because they don't have imaging at that level for humans.
Excellent. Over here.
I've enjoyed your talk. How do you think about cause versus correlation with NOS1-OP, and could it be the biomarkers the consequence of a lifetime of schizophrenia, and is that still useful?
Right, so we say there's correlation. Again, these changes to the DNA in NOS1-AP confer risk. They're not causative. So the problem with schizophrenia is there's really nothing that is causative. Everything is pretty correlative. And even the risk factors, I mean, you can say, okay, they confer risk. They cause risk, but they don't cause schizophrenia. If there's a strong enough correlation, I think that we still will be ahead of the game and ahead of where we are now. But this is part of the problem with studying the brain. It's very complicated. It's never very simple. That's why it's fun, right?
There's a question in the back, and I believe the last question was over here. Was there a question?
Hi. So you talked about a little bit like the difference in like gender between like men and women and like the effects like that medication differs. So within the Asian population that you see like NOS1-AP, have you also seen differences between like men and women within the Asian population?
That's great. So we only have 6. We have an NX. So even though we have females and males, we really can't do a statistical analysis. So that's why I'm applying for another internal grant to get more people. But it is something we want to know. We have different subpopulations, and because they're subpopulations, the N values are just too small to do those.
Excellent. And our final question over here.
Hello. Okay. So you talked about the fact that you can't actually like take any of these nerve cell samples from live patients. But are there not other nerve cells that you can kind of tap into? Like we have nerve cells throughout our entire body.
So part of the problem, so there's a better way, kind of a better way to go. You can't, neurons are all different. So you can't take a neuron from, let's say, the gut and assume it's going to act like the brain. What people do, it's not a perfect system, but what they do, and it's a little better, is they can take skin cells. People have taken, I think we could take cheek cells. We haven't started on that yet. We've discussed it in my lab. You can turn them into neurons. So many of your cells, like cheek cells, neurons, they're what we call differentiated. They've become what they're going to become. There's a way to take them back and say, hey, let's make you a stem cell. And then let's turn you into what we want to turn you into, assuming that the genes and the mutations and everything stay the same. There are problems with it, but people have done things like they've studied patients with schizophrenia, gotten more skin cells, not cheek cells, but skin cells, and then studied them and seen that they don't connect as well, that their dendrites look a little different. We even tried that and we saw their dendrites looked a little different, but we only had an N of 2. So people generally now, because of the technology, will make human, we call them induced pluripotent stem cell derived neurons, so that you take, cheek cells or skin cells or nasal epithelium and then turn them into the neurons and then study them that way. So you can drive them to look more like the neurons in the brain than if you took, let's say, a gut cell or something that's easily available, a peripheral cell or something, and it's less invasive. It's a really good question.
Thank you. So before I ask for a round of applause, will you be able to stick around a bit? I know folks will come up. All right, excellent. So if you have more questions, please come and let's thank our speaker. It's a lot of excellent work. Thank you so much. And thank you for coming.
Thank you for coming.
Thanks, everybody. And we'll see you all for our final talk next week.